Amazing boat. On we go. Oompa loompa. So Solar has a website. Solar at txbiomedgenetics.org. And it was asked during the break. It is my belief that all of these slides are going to be made available on the conference website. So everything you are seeing on the screen you should have in your hands at some point, and we can get it to you sooner if you need it before it shows up there.
Actually going to have a download option which includes all the scripts and all the code you see here.
Great, excellent. Thanks, Peter. All right, so Solar is available for essentially Unix-based systems. You need a Unix-based system to run it. You can freely download and install Solar from that website, but before it lets you maximize a likelihood, it will make you register. You have to write in for a key to the support email address. The reason we do that is that Solar is funded by a grant from NIMH, and your registering with us lets us provide a number of users to NIH to justify our funding and put in our progress report. We don't give out that Solar mailing list. We do not spam it. We send out an email maybe once a year letting you know of updates and bugs and so forth. So that is why we ask you to register and what we do with that information. This is also the support address for Solar how-to questions. You are better off emailing that support address than emailing me. While I am here standing in front of an auditorium talking to you guys, somebody is at home answering email to that address. So you are likely to get a faster answer by emailing the support address than by emailing me or John or any of us individually. Charles Peterson, who is one of the chief programmers on Solar, answers that address typically. Someone else covers it if Charles is on vacation. And if you ask a question that's really a theory question rather than a how do I force the program to do what I want question, Charles knows which of us to kick the question off to get you the answer that you need. So you've installed Solar, you type Solar and it starts up, tells you what version it is and so forth and so on, and you see this Solar prompt. So your prompt changes so that it says Solar. I mention that because when we run workshops where we have the opportunity to do hands-on exercises and everyone has a computer in front of them, invariably somebody makes the terribly embarrassing but common and understandable mistake that they are typing their Solar commands into the regular Unix command line prompt and they don't understand why they don't work. So if something you think should be working isn't working, your first line of defense is to check and see that you've actually got the Solar prompt and you're in Solar. Solar wants to be helpful. There's documentation for Solar available online at that website address that I gave you, but there's also a bunch available interactively from within Solar. If you type help, it will give you a list of all the Solar commands. It's scrolled off down the screen here, but if you scroll past that, it will then give you each command with a tiny one-line description of what that command does. So you can browse down that to see which of the commands might be relevant for what you're looking for. If you then type help and the name of any command, it gives you more information. So help and this XXX here is meant to be the name of any command. It will start giving you more information about that command. Solar accepts data files in two general sorts of file formats. PEDSys files. PEDSys is a pedigree database system that we have developed. It is not much used outside the Foundation. It's also freely available for download there. But since the other file format is just simple comma delimited text file, I'm just going to assume that you have no interest in investing in a new database system, and comma delimited text files would be just fine for your purposes. So there are 5 types of files that Solar has: pedigree, phenotype, marker, that's genetic marker, microsatellite or SNP, a map that tells about the locations of the markers, and frequency that tells about allele frequencies for the markers. Under help, it's So down here in the part that you can't see, in addition to the commands, there are also help entries for each of those files. So there's a help file-pedigree, help file-phenotype. Everything I tell you about the sort of contents and formats of these files is available both in the online documentation at the website and in those help file entries. So you can find all of this again. As long as you have a vague idea what it was you were looking for, I'm sure you'll be able to find it. The absolute first thing you need is a pedigree file. Without a pedigree file, Solr will not go. It will refuse to do anything for you. The pedigree file must contain one line for each individual and then a number of columns. So you must include an identifier for each person, their father, their mother, and sex. You optionally may include a family ID, an identifier to flag MZ twin pairs, and household ID. Household ID makes that house matrix I was talking about earlier. So you can put any numbered categorical thing in this household ID field. And Solr will take it when you load the pedigree file and make you a matrix of zeros and ones. Share, don't share. So although that's called household ID, you can hijack it to be an environmental sharing matrix for anything you want, as long as you want zeros and ones. Yes?
So if you want to add an extra field to that, you can do that? Or?
No, it will only do that once. So you have to be sneaky and load the pedigree once with your first thing, take that matrix, rename it, and then load the pedigree again with your second thing, take that matrix and rename it. So it's making you a matrix that it's outputting and storing. Doesn't actually, as long as that matrix is in the right format, it's not actually keeping track of it much beyond that. So you can make multiple matrices, name them multiple things, and use them. Or you can provide your own matrix that you've generated externally, as long as it's in the right format. And that format is, again, described in the documentation. And I think Peter will mention that a little bit. So you can make a matrix that has shades of gray rather than zeros and ones. As long as it's in the right format, you can name it house, and Solr will happily take it up and use it. All right, so I say that family ID is optional here. If you've given, for example, each person a number, I've got mother and father, 2 kids in each of my nuclear families, and I have not repeated IDs between families. 1, 2, 3, 4, I'm going on to the next family, 5, 6, 7, 8. You don't need the family ID. If the IDs are unique across the whole sample and only one person has number 2 in the entire sample, then you don't need to include the family ID. If you have repeated ID numbers across pedigrees, if this second pedigree is also 1, 2, 3, 4, and all the mothers are 1s and all the fathers are 2s and all the kids are 3 through something, then you need this family ID to differentiate a unique individual, essentially. Yes, Peter?
Perhaps you could mention that the ID doesn't have to be an integer number. It doesn't actually have to be a number at all.
Yep. You can use letters, you can use numbers, you can use combinations of letters and numbers. They don't have to start at 1, they don't have to go consecutively. You can use any reasonable sort of identifier that you want. So you do not have to, whatever study IDs you're currently using will probably work. You don't have to convert them into anything unusual to use them in this. Sex. Males and females, you can code as 1 and 2 or F and M. Obviously F and, did I say this somewhere? No. Obviously F and M, we understand, F for female, M for male. What about those 1s and 2s? It's 1 for male, 2 for female, and the key thing is it's the number of X chromosomes. That's how you can remember it to yourself. So females are 2s because they have 2 X chromosomes. That's the sort of coding scheme. MZ twins. If you have MZ, monozygotic twins, in your sample, you want to include the MZ twin field and give each pair of twins, each member of a twin pair, the same number in that field. Everyone who is not a twin can have either a 0 or a blank. So I'm using it to identify pairs of genetically identical individuals by giving them the same number. So the first twin pair each get a 1, the second twin pair each get a 2, the third twin pair each get a 3. You only need to do that for MZs. Dizygotic are treated as regular siblings, essentially. It's called MZ twin, but it's not actually limited to twins. We have one study that has a very rare set of identical triplets. You can give 3 people the same number in that field, and Solar will happily go along its way. It's just identifying a set of individuals who are genetically identical. So what if you don't have families? What if you have unrelated individuals? You still have this same basic ID father, mother, sex, but everyone has missing parents. That's all. You've still got a line for each individual. We've got our comma delimited text file, and we've just got some commas together here to indicate the missing father and mother, or, unknown as respect to our study, father and mother for each individual in here. Yes, you can. And there is value to doing so. The unrelated individuals can contribute to our estimates of the trait mean and variance and covariate effects. So the relevant commands here are pedigree load and pedigree show. You need to load the pedigree file, pedigree load, and then the name of the file. In this case, I called that file I showed you on the previous page unrelped. You can call your file anything you want because you're going to specify the name of it when you load it. It's a comma delimited text file, but as far as Solar is concerned, it doesn't need to have a .csv or a .txt down here at the end. You can actually have these fields in any order. You can put mother before father. That doesn't matter. Because you have at the top line of your text file the field names, essentially. So you're telling Solar what order things are in. For the pedigree file, it is looking for specific field names. So it's got protected field names for the pedigree file. The command pedigree show then gives you a summary of the pedigree data that has been loaded. It's a good idea to use that command to take a look at things and make sure it looks like what you think it should, essentially. There's an add-on to it, pedigree show all, which will give you detailed information about each pedigree, including things like whether it contains any loops, either inbreeding loops or consanguinity loops. This can be a way of catching data entry errors. If all of a sudden you have pedigrees with loops in them when you're pretty sure you don't have any incest in your sample, then it kind of hints that there's something wrong in the data file somewhere. So in this example pedigree, which has Sam, Bob, Joshua, Alicia, Sophie, Ralph, and Gladys, I've got 7 pedigrees, 7 individuals, 7 founders, because everyone's unrelated. The phenotype file is the same sort of structure. We have a comma delimited text file. We've got a line at the top that gives the names of the fields. This must contain ID and family ID if you need that to specifically differentiate your individuals. And then it can contain anything else you want, and they can be called anything you want, because you're going to interactively inform Solar which items to use for what. And it can be in any order, essentially, that you want. The crucial thing here is that missing data needs to be a blank. So Alicia down here told us when she showed up for the exam that she wasn't fasting, so we didn't draw her blood for the LDL because she finked out on our protocol. And that is represented by these 2 commas together here. A lot of programs use a 0 or a 9 or a 99 or a -9. The problem is, living in my quantitative trait world, all of those numbers could be a valid trait value for some trait that I can think of. So, missing data, blank. If you have yes, no, affected, unaffected sorts of traits, those should be coded as 1 and 0 or 1 and 2. Solar will not deal with letters in phenotypes. So you can't make it A and U for affected and unaffected. You've got to have a number there. Any phenotype or covariate that takes 2 values separated by 1 unit, Solar will treat as dichotomous. The phenotype commands sort of parallel the pedigree commands. It's pheno load and the name of the file. You can call it anything you want. I happen to call this one phenos. Stats-all is sort of the parallel to pedigree show. And what that will give you is, for each phenotype in the file that you loaded, the mean, the minimum, the maximum, the standard deviation, and so forth. And that's another way of checking whether the trait values that actually got into Solar are what you were expecting to see and that there weren't sort of data errors along the way. You can see it also gives you sample size here. I've got sample size of 5 for age and for smoking, but only 4 for LDL, because you remember Alicia didn't have an LDL value in our file. If you just type pheno, it will repeat that top line of the file back to you. So if you've forgotten whether you included something in this phenotype file or not, or you've forgotten what you called a variable, just typing pheno will get you the list. That works in this case if you've got 1,000 little data points that you have abstracted from your imaging data, then typing pheno and looking at what it puts out towards you might not be so useful. And Peter, who is talking next, is going to show some examples of analyzing imaging data and share some scripts and automation he's set up to deal with that situation where you've got thousands of traits. So from the commands that have been typed so far, Solar has created files that it leaves in the directory where you started Solar. Some of these are housekeeping files. From the pedigree file, it made files that it uses internally called pedindex and pedigree info. And it goes back and consults those to come up with the kinship matrix, which is that phi2gz down here, and to put out the stats that go with that pedigree show command. It's also got a phenotypes info file. You can ignore those files. You can open them and look at them if you want and poke them and see what's in there. But as long as you leave them alone, so Solar can find them when it's looking for them, you'll be fine. There are also files left that show copies of the results that were put on the screen. So all those stats here are sent to a file called stats.out. So you can go back and look at those results again anytime you want. So polygenic, the sort of classical aggregation of many QTNs of small effect is referred to as the polygenic model. And polygenic is the name of the command in Solar that gets you that heritability that we talked about earlier. So polygenic is tapping into that sort of thing. The polygenic model, basically you need to tell Solar which one of the things in the phenotype file is your focal trait of interest, which of the other things, if any, are covariates, and then run the polygenic with this optional -s for search. -s controls whether or not it does that likelihood ratio test for the covariates. If you've got your covariates, you're happy with them, and you don't want to mess with them, you just type polygenic. If you want to screen the covariates and get a p-value for whether LDL levels differ by smoking in this sample, then you want that -s on the end there. Once I've loaded that phenotype file once, I can go through this multiple times with different traits. So I can say one time trait LDL, specify some covariates, run my polygenic, and then come back later and say trait smoking and do a different analysis. Once you've loaded the pedigree file and you've loaded the phenotype file, Solar uses those housekeeping files to remember them. The next time you come back, if you come back next week and start Solar, it will still remember that same pedigree file and phenotype file as long as you're in that same directory where those housekeeping files are stored. So you can start at this point then, rather than having to go back and load the pedigree file and load the phenotype file every time. I think for this crowd, that's an advantage. If you've got thousands of potential imaging phenotypes, you only have to do that first step once, and then you can come back to your phenotype file over and over again and take different things out of it to analyze in different ways. So you specify the trait, you specify some covariates, you can specify functions of covariates. I can specify age squared. I can specify age by sex. The covariate command allows that. And there's a description of that under help covariate. And then this polygenic, with or without the S, runs through a series of likelihood models and ends in this summary of results. So this is still that toy LDL data set where we had the 4 unrelated individuals. So the heritability here is meaningless because we don't have related individuals. It's not estimating the heritability, but it's still forced to put it here on the output. We've got 4 individuals. We have significant differences in LDL levels by age and by sex in this sample. Once you analyze a trait, Solr makes a directory named after that trait. So in this case, it's made a directory called LDL, and it's left a bunch of files in that directory. A lot of them are housekeeping files, but these results that it's sent to the screen that you're interested in are in polygenic.out. So you can come back and find them again at any time. And that also means that if you're setting up a big batch script, like Peter's going to describe, to go through and analyze 100 or 1,000 traits, if storage space is an issue, you can be deleting, grabbing what you want from these files and deleting the rest. But if storage space is not an issue, you can come back tomorrow, later, next week, and go into the directory for a particular trait and find all the information for that. I want to point out down here on the bottom, it's giving a warning. Residual kurtosis is 1.2, which is too high. So that question of normality and the assumption of a normally distributed trait, Solr is looking out for you on that. It's checking the kurtosis on any analysis you do on the residual after accounting for age and sex, and will tell you if it thinks your residual is too high. You are free to ignore that and go on your merry way, but you are also free to pay attention to that and do something about it. Okay, so there's an important thing you need to know about those covariates if you're using the -s. What Solr does is it starts with a full model that has all of the covariates in there. So it's estimating a regression coefficient and a difference in mean trait levels for all of the covariates. And then it's dropping them each out one by one to do a likelihood ratio test, comparing a model where covariate 1 is forced to 0 to the full model. Get a p-value, put 1 back, take 2 out, get a p-value, and so on. This has an important implication. This means that if covariate 1 and covariate 2 are effectively the same thing, neither of them is going to turn up significant. Because when you take 1 out, 2's still in there. When you take 2 out, 1's been put back. And so neither of them is going to turn up significant. So you have to, if you care about the covariates, you have to be aware of that and choose intelligently in the face of that default. There's also a command called step forward that can be used to do it in the other direction and add covariates in one by one, starting from no covariates. In Solr, the sort of default analysis that most people are doing most of the time is highly automated. But it's very easy to go in there and mess with it and do what you want to do instead, essentially. All right. At any point, if you type the command model, it will show you sort of behind the scenes model that is behind the numbers it's been putting out to give you the results of your analyses. So behind these results here of that polygenic run, if I type model, I can see that the mean LDL levels in my sample is 154. It's increasing with age. Paradoxically, my LDL levels appear to be higher in females. So beta sex is the displacement of the trait mean for females as compared to males. I've got my model. Yep.
So females coded as a 1. That's why you said.
Females coded as a 2 for 2 X chromosomes.
So
And you can also code it as M and F.
I guess I was just wondering why you assume that beta of sex is indicating that of female.
Because that is Solr's default. That's what it's doing. You can see the model for the trait mean is down here. And whatever you've put in there for sex, whether it's 1s and 2s or M's and F's, gets translated into a 0-1 female yes-no by Solr. So you see here it's got beta sex times female. That female is the 0-1 that was constructed from whether you were using 1s and 2s or M's and F's in the pedigree file. So that's a Solar default. And you can see that this beta for age is corrected by the mean age. So this mean up here is the mean for males at the mean age of the sample. And this beta age is for a 1-year deviation from the mean age of the sample. And we've got our omega here, was that variance equation I showed you earlier. We've got our additive genetic component, structured by the 2 times the kinship matrix, and our environmental component, which is structured by an identity matrix. That's 1s down the diagonal and 0s everywhere else. So you don't share your residual with anyone. It's a unique environment. Okay, what about that kurtosis flag that came up there? The way we can deal with that, the simplest thing that we tend to routinely do is to normalize the trait values. The command there is define. I can specify anything here. This is what I want to call my new trait value. And the rubric that normalizes it is called Inorm. So I'm defining LDL norm, which is some new thing, could be anything I want to call it, as being Inorm underscore and the name of the trait I want to normalize, LDL. So you can do that to any trait and replace this with the name of something in your, any quantitative trait. Replace this with the name of something in your phenotype file, and then this can be whatever you want to call the result. Inorm is just one of the things that define can do. Define can be used to create new things using pretty much any sort of arithmetic formula you want to put in there. So I can define new things that are functions of anything in my phenotype file. I can square things, I can add them, I can multiply, I can divide, I can take the natural log, and so forth and so on. But what is that Inorm doing anyway? It's essentially ordering the trait values and then computing for each trait value a quantile on a normal distribution and replacing it with that. If you've got 2 individuals who have exactly the same trait value, then they get an average of their, they're still ordered and they get an average of their positions. So if they went in with the same trait value, they come out with the same trait value is the important point there. But there's also a help Inorm in there that will explain as many details as you want on that. Yes?
So if this already does that for you, then we don't need to normalize that data before.
Correct. Solar can do that for you. And then I don't have it in here, but there's a command called residual. So back here, after I ran my polygenic, I can run a command called residual, and that will save residual trait values adjusted for age and sex. And I can then Inorm that residual if I want to. So you can do those sorts of trait manipulations and do the normalization before or after covariates all within Solar. Okay. Yes. Yes.
So you can imagine quite a lot of situations where that might not go very well. I mean, so isn't this rather a rough way for dealing with problems in kurtosis or skew? Wouldn't it be better to deal with those, I don't know, in SPSS or something than just put the log transform or the square root transform or whatever your data suggests is best for your data, depending on how it's failing the tests of normality, and do that, and then put it in, or even if your command allows you to do it, it just seems like this might be encouraging people to do an uninformed transformation without knowing why they're doing it.
Yes. It may be taking a hammer where something more subtle would suffice. If I'm looking at 20,000 gene expression phenotypes, I'm not going to be able to go through and make an intelligent decision about each one individually. And so squishing them through my pipeline and smacking them with my hammer might be the efficient thing to do, and then whatever comes out the other end of my analyses, if there are 5 things that are interesting, go back and look at them in a more informed way. If you've got one trait that you are that you're the expert in and that you actually care about, you can do those square roots and logs with that defined command. Or you can do it in SPSS, and I mean, if you're comfortable with that, then do it there and import it in, yeah.
Actually, from my experience, I found that Inorm, which stands for inverse Gaussian transformation, is actually quite robust. And in fact, I was trying to be an informed person, and I was trying to apply a particular type of normalization to my data that's specifically benefiting it, and it was like a square root and logarithm transformation that I used. But when I actually compared the residuals, I found them essentially the same. So if you actually have a pretty well-defined transformation, Inorm will likely to mimic it and produce basically essentially the same results.
And have you looked at using quantiles versus using sort of, I don't know, sort of temps or whatever? Can you have that flexibility to say, so at the minute it's splitting data into quarters, right? Is that right? From what Astra and Edible is doing. It's allocating it based on
Inverse normal cumulative density function.
Yeah, it's inverse Gaussian transformation.
Right. So you could do it for finer grade.
So the marker file. Again, common delimited text file, line at the top that tells you what's in there. Here, you want to have ID and FAMID if needed, and genotypes only. And this is because, particularly when you have a GWAS of hundreds of thousands or millions of markers, you don't want to have to instruct Solr what to do with each of them one by one. You want to be able to say, this file contains genotypes, please take it and do the same thing to all of them, without having to specify them individually. So really, for purposes of efficiency, the marker file is set up so that it contains ID and genotypes only. You can have microsatellites in the marker file. I'm assuming that most people are not working with microsatellites anymore, but if you are, you can do that, and help file marker will tell you all you need to know. Something I haven't said is that if I've got somebody in my pedigree file that I didn't genotype for whatever reason, or their genotyping failed or something like that, they don't need to appear in my marker file. I can just leave them out. Leaving someone out of someone who's in the pedigree file being left out of the phenotype file or the marker file is just fine. If someone appears in the marker file or the phenotype file but not the pedigree file, they won't get used, and that's problematic. So you want to have everybody in the pedigree file that you'll potentially use ever. But you can leave people out of the phenotype file or the marker file. You also don't need to have the people in the same order in each file. So the information from different files is being matched up on the basis of this ID. I'm going to find Bob in each file, whether he's the first person or the third person or the 275th person in each file. You don't need to order the individuals in any particular way. So for purposes of example here, I have 3 SNPs. The SNP-specific commands I'm going to show you assume that your marker has 2 alleles and will balk and refuse to go if that is not true. So parallel to the other commands I've shown you, we've got snip load and the name of the file. You can call it anything you want. I called mine my snips, and snip show. And in this case, this is the little file I showed you here. It's showing me that I have 3 markers. I've got 7 people typed for each marker, so I've got 100% typed. And it's giving me allele frequencies that are based on just counting as it comes in. So these allele frequencies are just counts. They're not corrected for pedigree structure if you have pedigrees. You can get allele frequencies corrected for pedigree structure. You can get maximum likelihood estimates of allele frequencies taking into account the pedigrees. The command for that is freq, F-R-E-Q, freq MLE. And you can see again at the bottom, Everything that it's shown you on the screen that it thinks you might want later is saved in an output file. The map file is an exception to the rule. It's a regular old text file, but instead of a comma in here, it's just got a space. I don't know why, I don't know who decided that, but there it is. The exceptions, prove the rules. Again, you've got a header line, and then you have a line for each marker with the name. That's that same name that was in the genotype file, and a position. Because you can use this same map file for linkage, this line at the top specifies whether you're giving it a file with locations in base pairs or a file with locations in centimorgans for linkage. So my SNP-specific commands. After I've loaded the SNP file, which I did back here, and taken a look at the stats, the command snip covar takes those genotypes and makes that 0, 1, 2 covariate of how many copies of the minor allele does each person have. So that's just translating the genotypes into that additive genetic covariate. You probably want to use the optional no haplos on here. If you've got family data and phased data, you can use haplotype information in constructing those covars, but that is rarely the case. Yes?
Where do I specify the chromosome?
You can do that back here. It's not really used in the association analyses. Or you can put it in the name of your file. You can call it mysnips1, mysnips22. That's an excellent point. That is not yet taken into account. So this SNP LD will calculate an R² between all pairs of markers in the file. And it's, as you've correctly noticed, I can put SNPs from different chromosomes in the same file. And Solar won't care. So this command will calculate a correlation between the SNP, all of the SNPs, regardless of what chromosome they're on. That might be entertaining and informative, but it might be not what you want. So in that case, I would make a marker file for each chromosome. Oh, and that's another point. As distributed, the limit on the number of fields in a file is 40,000. So you're going to have multiple files for your GWAS anyway. It is possible to change that limit if you want to. It gets unwieldy, but it is possible. So this SNP LD calculates that correlation among those 0, 1, 2 covariates, essentially. And it gives you the R² measure of LD between markers. If you use the optional plot command, it will make you a plot that looks like this, where it's giving you a heat map with red being higher levels of LD for all pairs of markers. You don't actually have to use the map file with this if you don't want to. For the standard association analysis commands, as I said, this SNP LD is ignoring chromosomes. The only place that position is coming in is drawing this little location schematic down here. So if you don't specify a map file and load a file of SNPs, it will just essentially number them in order and consider that their locations. And the only result is that you're not going to get this little, an appropriate schematic down here on your LD plot. Another useful command is the fnum modifier to the SNP command. What that does is it takes these LD values between all pairs of markers and tells you, estimates the number of effective markers given the LD structure. So I've got 3 SNPs. Given the LD between them, the effective number of SNPs is 2.99. So my 3 pretend SNPs that I made up back here are not in strong LD with each other, surprisingly. The simplest way to run association in Solar is the mgesos command. When you run this snip covar, it leaves you a file called snip.genocov. And that contains a line for each individual with their ID and a field for each marker with, instead of a genotype, a 0, 1, 2 for how many number of the minor allele. So you can point to that file that you've just made, run the mgesos command, and it will run that simple test of do the mean trait values differ by genotype for every marker in that file. If you want to do a GWAS, you divide your genotypes up into an appropriate number of files and just do that 10, 20, 100 times depending on how many markers you've got in there. And again, I think Peter will show some scripts for automating that kind of thing. The output of this mgesos command looks like this. It gives you the name of the marker, a chi-square, because you remember we're doing a likelihood ratio test. That's a chi-square with 1 degree of freedom, and a p-value for the question, does the mean trait values differ by genotype at this locus? Is that regression coefficient different from 0? Yes? Yes, so they could still be correlated. There could still be gametic disequilibrium between them, if not linkage disequilibrium. But typically, I would not make this plot for markers that were on different chromosomes. Okay, so it's giving you a p-value for each SNP, giving you that beta. So I can say here that mean LDL is going up with the minor allele, and here mean LDL is going down with the minor allele. Let's see. For some reason, I'm not getting a standard error on those betas, maybe because I have a sample size of 4. I've got a variance explained, and I have down here the number of individuals who went into that analysis, 4. And again, it's leaving me an output file called mgesos.out that has everything. It's put to the screen here so I can come back and find it. So it's got that measured genotype model, testing whether the mean trait values differ by genotype, assuming that additive model of interaction. If you have families, there's an additional command called Cutie LD. You have to set it up with this Cutie LD cove. That sets up a file called snip Cutie LD cove. That's a similar file of covariates, but it's setting up the covariates necessary to run quantitative trait TDT. Then you can run the command Qt LD. That runs the measured genotype and also QTDT and a test for stratification. I will speak to those in the know because there's not time to cover everything, but that test for stratification is essentially looking at, so the QTDT has a within family component and a between family component, and is testing a within family component to protect you against stratification. The stratification test in there is comparing the within family to the between family component. In theory, if there's no stratification, those two should be the same, should not be different from each other. So you can run QTDT in Solar. Okay, Solar has a couple things that will make your life easier. You can use most Unix commands from inside Solar. So if I want to look at what was in my file, I can use more or cat or whatever without having to exit Solar. I can use VI or Emacs or whatever my favorite Unix editor is to edit my file and fix something on the fly without having to exit Solar. The exception is you can't use wildcards from inside Solar. Peter is going to talk about this more, but you can make TCL scripts, tickle scripts, to automate any series of commands and to build your own custom sorts of analyses. The command to script within Solar will build a TCL script for you out of a subset of commands that you have just run. So you can specify to script and tell it, I want to keep the last 4 commands I just ran and put that in a script for me. That essentially makes those last 4 things you just ran into a single command. So if you've got stuff you do multiple times, you can automate it, essentially. So, power users. You can directly specify the mean and variance equations. Everything I've shown you is set up to automate the typical things that typical people typically do. But if you want to, you can go in and directly specify the mean and variance equations and then use the command maximize to maximize the likelihood for that model. Keep that likelihood, change something to 0 or change a couple things to 0, maximize that, keep that likelihood, and perform your own nested models and likelihood ratio tests. So you can if you want to invest enough time to understand how to do that, you can make Solr do any sorts of complicated models that you want to do, essentially. Okay, one slide about linkage analysis, as promised. To quote Monty Python, linkage analysis is not quite dead yet. Obviously, it requires families. The basic structure for conducting linkage analyses in Solr is that first, you have to obtain maximum likelihood estimates for the allele frequencies, accounting for the fact that the allele frequency estimates you have when you load the marker file in are just counting. And it's not an independent observation that a child has the same allele as their parent. You have to calculate and store those estimates of identity by descent sharing that we talked about earlier. The good news is that you do this once, you store it, and then you can use it again and again and again and again to analyze your 2,000, 5,000, 10,000 imaging traits. The bad news is that IBD calculation can be computationally intensive and time-consuming. The larger your pedigrees, the more computationally intensive it is. So unfortunately, the exact sorts of pedigrees we just told you would give you more power, also make your analysis time longer. Then the commands that actually run the linkage analysis are 2-point or multipoint. 2-point is using IBD matrices that are generated from a single marker. There used to be conceptual reasons that people advocated doing that because of imprecision in our marker maps. We're not so much worried about imprecision in our marker maps anymore. So typically you would want multipoint, which uses IBD estimates that take information from all the markers in a region, giving you a better, more precise estimate of IBD. Solr will calculate multipoint IBDs for you, but the routine that's in Solr to calculate multipoint IBDs was state-of-the-art for 1998. It was the only thing in 1998 that would let you have as big a pedigree as you could make and as many markers as you wanted. The field has moved on since then. So we do not use Solr's multipoint IBD estimation routine to do our multipoint linkage analyses in Solr. We actually use Merlin, Gene Hunter, SimWalk, or Loki to calculate IBDs and then import them into Solr, depending on the size of the pedigree. Things like Merlin and Gene Hunter are exact, but there's a limit on the size of the pedigree you can put in there. SimWalk and Loki are Markov chain Monte Carlo based methods. They take longer, they're not exact, but they can handle that 2,000 person pedigree from Nepal that I might have mentioned earlier. So, if you are interested in multipoint linkage, email that help address and you'll be put in contact with Tom Dyer, who will give you the scripts to get multipoint IBDs out of one of those programs depending on the size of your families and import them into Solr. We will help you with doing that correctly rather than using the antiquated, outdated, state-of-the-art item that's actually in Solr. Okay, recap. You can get Solr at the website. That's also where a bunch of documentation is. And there's the user support address for questions and the Solr registration key. That was the end of what I had. Next, Peter was going to talk about imaging data specifically. And then David has a little bit on IRVs and some examples and interpretation. Should we have a break?
Yes, a 5-minute
5-minute
break.
break? Okay.
There are so many patients